The scan came back clean. The problem was what it said. In 2019, researchers sequencing ancient DNA from a 10,000-year-old skeleton at Cheddar Gorge, Somerset, England, ran the sample through pigmentation gene sequencing. The result was precise. It was also, by the standards of the model science has relied on for 2 decades, essentially unclassifiable. The genome carried neither the dominant brown-associated alleles nor the blue-associated alleles that modern predictive frameworks say should be present this early in European prehistory. It carried something in between, intermediate variants, distributed across multiple loci, producing a signal the dominant model has no clean category for. And that skeleton has a face. You've seen it. The Cheddar Man reconstruction, dark skin, light eyes, that image that circulated everywhere in 2018 was built using the same model that just failed to classify its own source genome. The eye color in that portrait was assigned by a framework that, 1 year later, couldn't explain what the DNA was actually saying. Hazel eyes have no agreed scientific definition. None. The framework governing how science predicts eye color from DNA was formalized between 2008 and 2012, anchored by a landmark paper in Nature Genetics by Iberg and colleagues identifying a single genetic variant near the OCA2 gene as the primary driver of blue versus brown eyes. The model is elegant. For the extremes, it works. When the allele at that position runs one way, you likely get brown. When it runs the other, you likely get blue. Predictive accuracy for those two outcomes sits around 74%. That number became the foundation of an entire field. The model was built on blue and brown. Hazel was an afterthought, and that is now a problem. If you have hazel eyes, the most advanced forensic DNA tools currently available cannot reliably predict your eye color from your genome. The model doesn't have a category for you. It has a placeholder, the word intermediate, which sounds scientific. What it actually means is the system doesn't know. It just doesn't say that out loud. Think about what that means. Police agencies across more than 30 countries use OCA2-based prediction tools to generate suspect descriptions from crime scene DNA. Composite sketches are shaped by those predictions. Witness accounts are checked against them. Hazel eyes appear in roughly 5 to 8% of the global population, up to 25% in parts of Europe. Every one of those cases runs through a model that returns an answer it isn't equipped to give. The researchers who found the anomaly in the Cheddar Gorge genome didn't go public immediately. They went looking for more samples. The model hadn't been wrong before. It had just never been properly tested on Hazel. The tool at the center of this is called IrisPlex, developed by Walsh and colleagues in 2011. It was a genuine scientific achievement, a compact, validated instrument capable of generating eye color predictions from trace DNA, partial profiles, degraded samples. Within a decade, it was deployed across 3 continents. Its accuracy figures are real and published, but IrisPlex was trained and validated primarily on Northern European populations, with high frequencies of clearly expressed blue and brown phenotypes. Hazel-heavy populations across Southern Europe, the Caucasus, Central Asia were underrepresented in the training data. The published accuracy rates measure a narrower problem than the one science was claiming to have solved. But the real problem wasn't what IrisPlex got wrong. It was what it couldn't even attempt. It doesn't have a Hazel category. It predicts intermediate. That is not the same thing. The scale of that gap became undeniable in 2021 when a large genome-wide association study identified more than 50 genetic loci influencing eye color. Not 2, not 8, 50. These loci include genes regulating the density of the iris stroma, the distribution of melanocytes across different layers of the iris, and the presence of lipochrome pigment, a yellowish compound responsible for the greenish-gold shift that makes hazel eyes change appearance under different light. No simple melanin model predicts lipochrome. The 2-gene framework captures roughly 20% of the genetic architecture of eye color. The other 80% was sitting undescribed in the genome the entire time. 50 loci. The textbook said 2. The question isn't just how that gap existed. It's how it survived peer review for a decade, while the predictions built on it were being used in courtrooms. What does intermediate mean in a forensic report? It means we don't know. It just doesn't say that. 3 independent research groups arrived at the same structural problem from different directions. The UK Biobank Pigmentation Studies, the Estonian Biobank, and a 2022 reanalysis of Northern European ancient DNA by Sisa Parnoja and colleagues each identified the same gap between what OCA2-centric models predict and what human iris variation actually looks like across diverse populations. None of their datasets overlapped. All of their conclusions pointed to the same incompleteness. 3 labs, 3 datasets. One conclusion they all avoided naming directly. None of them called Hazel a distinct genetic category. All of them treated it like one. There's a reason the problem runs deeper than genetics alone, and this is the part the model was never designed to handle. The melanocyte, the cell that produces melanin, does not behave in the iris the way it behaves in skin. Iris melanocytes don't migrate through the same developmental pathways. They don't respond to ultraviolet radiation the same way. The iris stroma, where much of Hazel's variation is expressed, appears to operate under a partially separate developmental logic from epidermal pigmentation. The OCA2-centric model was constructed by analogy to skin color genetics because that was the better understood system. The iris is not skin. The model was built as if it were. This matters geographically as much as genetically. When you map Hazel eye frequency across living populations, the distribution isn't random. Hazel concentrates along specific corridors: the southern European coast, the Caucasus, Central Asia, the Zagros Mountains. These are not scattered occurrences. They trace the same paths carved by 2 of the most consequential migration events in European prehistory: the Anatolian farmer dispersal into Europe beginning roughly 9,000 years ago, and the later Yamnaya expansion out of the Pontic steppe. The geographic clustering of Hazel overlaps with the genetic signatures of those movements in ways that are too structured to be coincidence. 1 person in 25 in Ireland carries Hazel eyes, 1 in 200 in East Asia. That is not noise. Selection does not favor traits randomly. If Hazel represents a distinct phenotypic output with its own partial genetic architecture, and the evidence increasingly suggests it does, then something was driving its preservation and spread across specific corridors of the ancient world. What that something was remains genuinely unknown. Links to low-light visual sensitivity have been proposed. Associations with other traits under selection pressure have been discussed. Pleiotropic effects through shared pigmentation pathways are possible. None of these are established. What is established is the distribution, and the distribution demands an explanation the current model cannot provide. This is not the first time a trait science considered simple turned out to carry a hidden architecture. Lactose tolerance spent decades as the textbook example of a clean, well-understood genetic adaptation, a single mutation spreading through herding populations, a satisfying linear story. Then the ancient DNA came in. Between roughly 2002 and 2007, a series of population genetic studies revealed that lactose persistence had not spread through one mutation moving cleanly across populations. It had evolved independently, multiple times, in different geographic locations, through different variants in the same gene region. The dietary evolution model had to be substantially rewritten. Not because the original researchers were careless, because the original datasets were incomplete. And incompleteness looks like certainty until the sample size gets large enough. Science revised lactose tolerance when the ancient DNA came in. The ancient DNA for eye color is coming in now. The question isn't whether the model will be revised. The question is what the revision will cost. The criticism of the 21 findings has been substantive, and it deserves acknowledgement. Methodologists have raised legitimate concerns about phenotype self-reporting. Studies that ask subjects to categorize their own eye color introduced real classification noise. Because Hazel is defined inconsistently across languages, cultures, and individuals. Sample sizes for Hazel specifically remain smaller than those for blue and brown, reducing statistical power for the intermediate range. Replication across ancestry groups has been uneven. These are genuine limitations. They address methodology and sample composition. What they do not address is the core finding, that a documented, measurable gap exists between what OCA2-based models predict and the phenotypes that exist in human populations. The criticism addressed the interpretation. It did not address the gap. The anomaly survived every objection raised against it. The stakes of that survival extend far beyond academic genetics. Ancestry platforms that reconstruct your physical appearance from a saliva sample. Forensic databases that generate suspect descriptions from trace DNA. Ancient face reconstructions that assign specific eye colors to Neolithic individuals and present them as scientific visualizations of real people. Every one of these systems inherits whatever assumptions were baked into the underlying pigmentation model. The Cheddar Man portrait, the most widely reproduced ancient human reconstruction in recent British media history, was built using OCA2-centric predictions. The Neanderthal faces in major natural history museums use the same class of model. The eye colors in those images were not measured, they were predicted. By a framework that its own creators now know is incomplete. The model did not fail spectacularly. It just quietly stopped working at the edges. The edges where most of the interesting history happens. What sits at those edges is something physical and specific and irreversible. The Cheddar Gorge genome is not a theoretical problem. It is a set of allele calls from a real individual who walked across what is now Somerset 10,000 years ago. The intermediate pigmentation variants in that genome have been sequenced cleanly. The data is complete. Under current models, the result returns intermediate, the system's formal acknowledgement that the prediction cannot be made with confidence. Not because the genome is degraded, not because the sequencing was poor, because the interpretive framework built to read it was designed for a problem slightly different from the one this genome is presenting. We have the DNA. We just don't have the theory to read it. And here is what makes that permanent. The iris does not fossilize. There are no preserved cells from ancient populations, no tissue samples that could be examined directly, no optical record of what Mesolithic Europeans actually saw when they looked at each other. Phenotype, in this context, can only ever be inferred from genotype. And the genotype to phenotype pathway for eye color is, at this precise moment in scientific history, demonstrably and measurably incomplete. Every ancient face ever reconstructed and placed in front of a camera, every Neolithic farmer, every Bronze Age portrait, every deep-time human brought back from bone and DNA, was given its eye color by a model that cannot fully account for what sits between blue and brown. Somewhere in a sealed archive in Bristol, the Cheddar Man genome sits fully sequenced, fully preserved, the most complete ancient British DNA ever recovered. And the one thing it cannot tell us, with the tools science currently has, is the color of his eyes. Not because the data is missing, because the key hasn't been built yet.